water if I want some.
Oh, we need
I have bottled one.
Yeah.
But,
I'll just try to. No, the water is fine.
no, sorry.
So take it.
No, I was just saying that in my bag I have a bottle that was filled with the water that is just here in this dispenser, water dispenser. Anyway, if he can bring a glass with water, that's fine. Yeah.
But you can use the water,
Yeah, the filtered one.
the filter water in your room.
Yeah. No, I tried, and it's saying like I used it and it worked, saying like it's drinkable.
It's good. No, it's good. So you did a postdoc with Martin, right?
So, yes, formally, I would say yes. Informally, because formally that was doing my PhD, so I was supervised in Krakow by Professor Zdzisław Burda, but the project was jointly with Olivier Martin, so you could say he was a co-supervisor, and I spent like almost a year in Paris.
Same university or
No.
different?
So, sorry, so my supervisor, yeah, the same university. Now he moved to a different university, and I moved back there.
But you did your PhD in
Yes,
Jagiellonian?
yes, Jagiellonian University, yeah, and then I moved for a postdoc to Institute of Science and Technology, Austria, and worked.
Austria.
Yeah.
Where
But-
in Austria?
So it's IST, so it's close to Vienna. Formally, it's close to Neuburg, but back then it was just established, or 5 years after establishment, and they attracted a lot of good people. Like,
So when we had our first Zoom meetings, that you were still in Austria.
yeah, if that was no, that so I moved to Krakow like 5 years
You're
ago.
already involved.
So like I don't know.
So
PhD.
when you had given a talk in
Yeah, I was in Krakow.
you were okay.
Yeah, I was in Krakow because that was like what 3 years ago.
Right.
I think so. And,
So we'll ask them to put one over there.
yeah. Thanks.
Thanks. So just before COVID, you moved to Krakow.
Yes, we actually managed to do all the moving before COVID, so we were kind of happy because I
So
know.
this is the end of 2019.
Yeah, so end of yeah, exactly 2019 I moved, and then and then we established, and then the COVID came,
Yeah.
but.
But at least you managed to establish.
Exactly, because I know stories from other people, moving during this period, and that was crazy. Like, yeah.
All right, so welcome to the last session for today, and for this session we'll have Professor Marcin Zagórski, who's going to give his first talk, Processing of Biological Information in Development Systems. So Marcin, we have known him for almost 5 years now. So he gave an online talk at IMSC just before COVID, or was it during COVID? I forget.
I think in COVID,
Yeah.
because it was online.
Right. So he did his PhD at the same university where he is now, Jagiellonian University in Krakow. Then he moved for his postdoc at Institute of Science and Technology in Vienna, and then 2019 he moved as a faculty member of the Jagiellonian University in Krakow, and he's been working on a number of very interesting problems in pattern formation. So we are very, very lucky to have Marcin fly all the way to India just for giving talks to us.
Thank you, Sitabra, for introduction. I always say that when I think about giving these online talks and then meeting people there, that this is often like you just sit in your office, you log in, then you log out. But in this case, I think you are the only example of a person that I actually interacted during such a talk, and then we continued, and then I was invited here, so I came with a pleasure. And yeah, so these online talks are not like completely waste of time. If you have some, it might work out. Today, I'm going to talk about how biological information is processed during development, and I will focus on positional information, biological positional information. I will define this later on. It's going to be pedagogical talk, so also feel free to interrupt me. First, I will try to give you intuition, and then, yeah, basically give you the framework of how you should define, how you should then use, and what it can give you, the framework of processing of information. But before I start this positional information part, let me start with a simple yet, I think, more fundamental question. How is it possible that from a single cell, a multicellular complex organism develops in a very precise and reproducible way? It all starts with a single cell. The cell quickly divides, and the cells increase in numbers. Here you see an early stage mouse embryo where two daughter cells were labeled with different fluorescent markers. At this stage, they're very similar. So if not for the staining, they would be indistinguishable. Yet, already 10 days after fertilization, you could appreciate that the embryo develops sophisticated structures, organs develop, body parts develop. Here, I think, yeah, blood vessels are in green, the nuclei in blue, and actively dividing cells in red. And for me as a physicist, you could think that our cells contain DNA. This is some kind of information that is then unwrapped to create such an organism, and this unwrapping requires energy. So you pump energy, you get this system to form. But I will make this question of processing biological information more specific. So another thing that is amazing here is that cells know exactly when, where, and what biological function they need to perform. So how cells access this information? What gives them this information about their position in the tissue? And the answer is they have access to a coordinate system that is created by morphogenes. Morphogenes are signaling molecules produced at the specific source regions in the tissue. So we can imagine here there is some source of morphogen. And then the signaling molecules diffuse or spread through the tissue through different mechanisms. We had this in the last talk. And then they create the gradient of concentration, often exponential, due to diffusion and degradation. And then this concentration is read out by gene regulatory networks that are in each cell. So here you can imagine a tissue, and at every cell there's gene regulatory network that interprets the concentration of the morphogen. You could say, this is really simplified because on every cell there are some receptors. The morphogen ligands are being bound to this receptor that trigger signaling activation pathways. There are some effectors, but at the end you could say there are genes that if this morphogen concentration is high, let's say, blue gene is expressed in the cell. If the concentration of a morphogen is intermediate, white gene, and if it's low, it's the red gene. So we recover this French flag. And this pattern of gene expression, the stripes in this pattern give rise to different body parts and organs. But if we would like to, so that's one way of representing this, how this positional information, so cells in this pattern, based on the concentration, could orient themselves in the tissue and find and establish a specific cell fate. If we now want to make a link with this framework of positional or basically processing of information, we say this is the input signal, this is the output signal, and what decodes the information contained in the input is a gene regulatory network. If you want more maybe pedagogical representation, you can think about this as some kind of GPS, or global positioning system. This is something that reads the position, based on the signal, it finds where you are. And then there is, so here I have two. So one is the position in the tissue and the cell fate. So this is Polish ID. This is how it looks. Because in this framework, you don't have direct specification of the cell fate in a sense that you don't know which genes are expressed. It's more like you have a position and at that specific position you observe cells that have specific cell fate, and then you could relate one to the other. I will talk a bit more on that later on. And now let's imagine that we don't know gene regulatory network, so we don't know the decoder. But we still would like to know how to get from the input to the output signal. And the idea is, let's assume that whatever is doing this decoding is doing it optimally. Saying this information decoder tries to use all available information that is in the signal, useful information, to get the output pattern. And let's see whether we could make some predictions with that. Because if you construct the network, you could then ask, okay, I knock out some genes, I have some mutants, and then I predict a new pattern. But without the network, can we do this prediction? Saying like we construct such a decoder, and then we do also predictions for the mutants. And now for several slides, I will try to convince you that it's possible. And one of the best studied systems where this was shown is the Drosophila. So this we already seen a few times. So again, maybe just saying that bicoid at the head side, anterior maximum, then it gets this really nice exponentially decaying gradient. But what I would like to focus on is actually the downstream. We also had this, like a talk before, the so-called gap genes, and I will use their expression as the input signal to define the output signal, which are parallel genes. And again, just giving you an overall view on how this positioning information that is, okay, first this anterior, this is bicoid, this is another maternal signal, nanos, that is more posterior, and there is also the third one with T. And this is all based on a for me, very neatly written paper by Petkova and others in Cell. So if you want to know details, I refer you to this one. So here there are 3 gradients. Then they specify this gap gene, 4 gap genes, Knirps, Hunchback, Cripple, and Jant. And then those, through some enhancers of other genes like IF, specify the stripes. So we will try to see how you can take this as an input and get this. So now the question is how to construct such an optimal decoder. You start with input signal that is that we denote by GI, which is the gene expression level. Here, X goes just marks the position in the tissue, and G1 would be the cripple, hunchback giant, and NIRPs. And then one more assumption is that we assume that at the given position, this distribution of the signal is multivariate Gaussian, which is mostly technical assumption because it's much easier to construct a smooth distribution with fewer measurements than just have a lot of measurements to really construct a full distribution where this is the mean of the signal at the given position x, and this is the covariance matrix. So this is, you can think about this as Gaussian, but just in higher dimensions. And now, a key observation is that you want to get the distribution of the fate given the signal, saying like the cell doesn't have access to the actual position. The cell sits in the tissue and it has access only to the concentration or gene expression, so the signal. So you want to know what's the most likely fate, or in general, what's the distribution of this implied fates for a given signal concentration. And to get this, you apply the well-known bias rule. So this is your pure distribution. Initially, you don't know anything about the position. So this is like uniform. The cells, without any signals, the cells, you assume they're uniformly distributed, or their fate. Then this is the measured distribution of the signal at every position. And then this is normalization factor. I will, if you, I will do it step by step on the next slide, so no worries. If you don't follow, this is like a sum up of the theory. And then, if you construct such a decoder, it's guaranteed to extract all information that is that any cellular or computational mechanism could extract from input signals. And further, if this is for a given concentration, it gives a specific peak, saying like unambiguously, one can do another substitution, saying like now we take a signal that is measured in the wild-type embryo or mutant embryo. So at every position you measure a signal, and using this distribution, you just put it here, and then you would have for a given position in your mutated embryo or wild-type embryo, you have a prediction of what's the probability of the implied fate. And again, let's see how it works on the example. So just one more thing to say about when I say the code is good or unambiguous. I mean that for a given concentration, you actually have this implied fate is sharply peaked, because you can also imagine different scenarios, and later on for this talk I will also give you some examples of that, not in Drosophila but in the spinal cord, where you have this so-called positional error. This is this sigma x that gives you basically your uncertainty, or the cell. You can imagine cells sitting here and trying to figure out based on the concentration what's its fate. So of course, if this input signal is really like broad, this uncertainty is high. And there are also possibilities that you don't have unimodal but bimodal, this distribution of implied cell fate. And formally you could define, that's your positional error, so that's the most likely or average fate, and then the spread around that would define the positional error. And just to make a connection with this picture that I showed you before, so maternal ingredients here, they act as some initial source of positional information. Then this is recoded into these 4 gap genes, and these are these GIs. And then these GIs are measured, and you need both the mean at every x, and also you measure, once you do more measurements, you could also reconstruct the noise and then reconstruct estimate the covariance matrix. You could then make this construct with the bias rule the posterior, And at the end, you get the decoding, where the idea of decoding is that if your predicted fate corresponds to the position of the stripe in the wild type that was used to construct this decoder, you predict there should be a stripe at this position. And now let's take, instead of 4, let's take one signal. So this is the Kyr, so cripple signal measured. This is the position in the embryo, and what you see is, for most, it's shallow, it's flat, then it's peaked. That's the image of fluorescent representation of the signal. And now, if you want to invert that distribution, you basically could flip it. And then, from the cell perspective, you ask, okay, now the cell, imagine cell here is something close to the anterior side, and it experiences a very low concentration of this signal. And there's, that's the probability of this cell finding itself in these regions, which is very broad. Then you go with the cell sitting in the middle of the embryo. It's actually much better, but still it's ambiguous. Okay, so cell could very likely be here or here, and only if it's close to the peak, it gets very nicely positioned. And now, this is okay. So that was what we had, and now you construct this decoding map. So it's like saying, for a given position, you give the implied fate. But what I would like to make clear with this picture, that there is, with such an input signal, there is just one position where you can unambiguously find that the cell could figure out its position. But now you add more signals, and what happens? This gets more and more narrow and close to the diagonal. Saying like adding one more, so hunchback, it already improves. Adding giant, it's even further improved. And finally, if you have 4, it's a very narrow reach of probability distribution that basically unambiguously allows cells to figure out their position in the tissue, hence their cell fate. But maybe it's not so surprising, because we used the wild type data, so no mutants so far. But and we tried to predict, based on this wild type signals that were measured, what's the implied fate. It's already somehow maybe interesting that it allows to achieve this very high patterning or precision, because it might be that the cells are, let's say you have 4 signals, but they are still not enough, then indicating that probably you need more, or that you're still using this 4, you couldn't reach this, but you see the stripes are positioned quite well, then it might indicate that still you are something missing. But in this paper, the authors asked, okay, now let's perturb these material ingredients and see what happens. Can we still do the prediction? Normally, you would need to construct gene regulatory network and make a prediction. Here, no gene regulatory network was constructed, just the decoder, which gives you these probabilities. And if you perturb the picoid, you just delete it, you affect the distribution of your input signal, and then, but you still apply the same decoder. So that's what you observe, or average over several embryos, and then you see that this probability reach of densities moved, and you could see that some of the stripes got duplicated, like this stripe got duplicated. Some of the stripes disappeared. they're just smeared. And you could say, okay, this is something you see here by just having a broader distribution in this probability density map. And it's just not this one, but they actually did a lot more. So I will just quickly go through those, don't explain it because it's not so important what changed there, but if you perturb 1, you can do this prediction. If you perturb 2, remove, basically, the embryo performs just worse, but that's probably expected. And then finally, if you remove all, you basically have no stripes. So you remove completely the information. And now if you quantify all these predictions, they found out that with very good, I think they predicted 95% of all possible intermutants, all possible shifts, disappearance, and duplications in this parallel stripes. So that's kind of convincing. They also had, I think, 3 predictions that were not fulfilled, and yeah, but still, it's a strong indicator that due to probably evolution and adaptation, this system in Drosophila really pushed forward or pushed this network that does this decoding to really be close to the optimal limit of processing of noisy signals. And now let me go a bit further and finally define what is this positional information. We are using this term, yes? Sorry. Oh, yeah. So these are different types of stripes, like if, the different genes expressed. And this is the measured peak location. So you have a mutant. That was the question? Yeah, okay. You measure the location in the mutant, and then what was the implied peak location. So, yeah, and yeah, I think that was. I should check what was the coloring, but I think that was. Oh yeah, I remember that was the distance. So if it was close to the predicted position, it was close to blue. But if it was a very far prediction, saying like you expect stripe, let's say at I don't know 0.7, but it reappears at 0.2, then that's why it's red. Okay. Any further questions? Okay, so let me move on. Yeah, so let's think a bit. Let's stop for a bit and think about this positional information thing. Intuitively, it's very hand waving. You could say, okay, there is some gradient, it contains information, but what is this information? So, in general, when you have a random variable x and another random variable y, then if you have some change in x and this allows you to make a prediction about y, then x has some information about y. Okay, so if x would be completely independent of y, then there would be no information between them. So Shannon identified mutual information as the unique measure that captures this statistical dependence between x and y. And he used, to define it, he used another basic concept, the entropy, defined here, which for this particular application, I would say the entropy, you could think about the entropy as a dynamic range of the distribution that's related to the variance. So you could also think of, okay, you could notice that 2, because we want to have information measured in bits, and it's like, yeah, average of log p. Then you define the mutual information between x and y as a sum of entropies for individual entries, and subtract the joint entropy of both, of the joint distribution. And if yeah, you could simply check that. If you put x and y for independent, this is then the product of px times py, and then of course this separates. So if you plug it here, you find this is 0. And just again, why this is more general than, let's say, the concept of Pearson correlation coefficient. That Pearson correlation coefficient here, maybe you have seen such kind of plots, I don't know, on Wikipedia if you Google Pearson correlation coefficient, because Pearson correlation coefficient just gets the linear relation. But mutual information gets you much richer statistical dependence, non-linear. So here, no dependence, irrespective of whether it's Pearson correlation coefficient or the mutual information, like here 0, 0. Here, linear correlations of both are giving roughly the same, but here the variance of the points is actually increasing with x, and then the linear correlation would give you still 0, and again the classic example where you have a circular cloud of points, you still get 0. But having said that, let's get back to position. So let's get back to mutual information, where you now would like to ask a question. Okay, how much positional information can be encoded by a single gene or some morphogen gradient or like gene expression? In the simplest case, you have 1 bit, or okay, the simplest would be just completely flat, no information. But let's say you want to have embryo to be able to distinguish left and right, or head and tail. And this is achieved by having high gene expression on one side, low at the other. This is 1 bit of information. And if you now, okay, one more observation, this g of x, gx is just a number. I get this definition, but it's a number that in bits that will give you, if you take 2 to the power of this mutual information, you will get the number of possible distinguishable gene states. And now you can think how to improve situation, how to get more out of the signals. Then of course you could make some intermediate levels of signaling available to the cells, and then if this noise is not too high, because if it would be too high, then you would still have very small change to distinguish between something that is left, right. Okay, here the high noise, but if it's too high, it could completely smear this difference between left and right. And this fluctuation is sigma g. This is your fluctuation for a given position in gene expression. Then, okay, this is the inverse of the slope. So of course, inverse of the slope means if this would be flat, then the inverse of the flat thing would give you very high number, so very high positional error, and vice versa. If it's steep, then the inverse would give you a very small number, so the positional error would be smaller. And you could even show that the optimal, or you could maximize this the number of distinguishable gene states if your input signal is linear and you narrow down this positional error by narrowing down the fluctuations. And of course, there are some biochemical limits. You cannot do it, indefinitely. In theory, you can, but in practice, you cannot. But let's see. I will also give you some connection, deeper connection between positional information and this positional error quite soon. And now, if you want to define it, or maybe not define, because it was defined, this is equivalent definition, but maybe somehow more useful when you think about this. So mutual information is, let's say, the useful part. There is also the total entropy. This is the distribution of the gene expressions. Or you can do experiment and measure how much, the histogram of gene expression, and then calculate its entropy. So it would contain both useful part and, let's say, something that I would call noise, pure noise. So then if you, from the total entropy, if you subtract the part that corresponds to noise, which is this one, which is again, this is the entropy of conditional distribution. This is something that you measure, and this means just the average of the signal. So this is simply I didn't put this here, but when I mean something like this, and in general x or px, this is just of whatever is here. Okay, and okay, so yeah, that's basically what I would like to, or maybe another way to understand this plot is that the cell without any input signal, it can be equiprobably at every position in the tissue, but after the signal is introduced, you actually narrow down the position of the cell. If this distribution is narrow, then the cell could position it with quite good precision in the cell. And yeah, this is basically restating that more formally. If someone would like to know more details, I refer to this paper, which contains a lot of mathematical framework behind this. But now, what we can do is, okay, like this decoding that we saw in Drosophila, you get the positional error, but like how is this positional information somehow useful, or maybe can we use it with in Drosophila? Because up to now I hadn't tell you whether how many bits has this these four signals that we were analyzing. So let's do it here, and let's ask, is this positional information conveyed in the input signal sufficient to explain the output pattern, the precision in the output pattern? And to do it, we need two more ingredients. So one thing is the data processing inequality, which is basically telling you something simple, that if you have some signal, then this is decoded, gives you another signal, and then this signal is further decoded, you don't gain information. Saying like this positional information or here mutual information will be always lower with each step. So that creates this dependency chain. So if you get from the position, you get the concentration. From this concentration, you get the implied position. Then if you have mutual information of how much signal gives you information about the position, this will be always greater or equal to mutual information of the implied position about the cell fate or the actual position. Also, this mutual information is symmetric, saying like you could always swap these two. So if we now plug it in, and again, I don't, all the details you could find in this previous paper. You could plug it in this definition that we had on the two transparencies before. You get this, And with this, you now could calculate it, saying like this is just the entropy of uniform distribution. So you could use the definition of entropy. It would end up with minus log 2, and this L squared gets from that. This term is a bit more tricky because we don't know what's the actual, in general, this probability, conditional probability of implied fate given the actual position can be some kind of complicated distribution, but we know that its entropy must be less or equal to the entropy of Gaussian distribution of the same variance. And then we could use this to get a lower bound on the information between true and implied position. And if we do that, and again, that's the expression for the entropy of Gaussian distribution. And if we have it both, we see this might look like a kind of complicated expression, but in the end, it's, those are constants, and what is here is the positional error, and if it gets small enough, saying like log of very small number will give a larger number, also with negative, so don't worry about the sign, and it means that if you would have large positional error, your mutual information will be low. But if it gets lower, this mutual information will get higher and higher. So if you now have a situation that one thing is to use the signal to estimate this mutual information, and the other is you estimate it from the actual position of the stripes. Okay, here I just showed that you could have equivalence between those two. This again might be a bit, I'm rushing through this, but it's just to tell you that, or maybe intuitively would be, imagine you have a very sophisticated signal at the start, but cells might use it, all available information, or they might not. For some reason, there is just more available information, and the cells are just using part of that, and they are still very imprecise, or they don't care so much. But this is not the case. And in this work, what Dubois and others, Gasper, Gregor, they show that if this number you get from the input signal, and this is what you get with this other formula. And this is very close to each other. So you get this 4.2, let's say, bits of positional information in the pattern in Drosophila. And okay, well, you could say, okay, but that's just a number. Can it tell us something maybe new? And one answer is that it shows that cells are using effectively this information. This is because these two numbers get close to each other. But the other may be more interesting. We heard a bit on Boolean networks, or how you make this different stable points corresponding to different gene expression. That if you would have just 0-1 expression of genes, saying like if you would consider just very low over very high, determining your cell fate. So here, what you have is the same system, but now you get the thresholds for distinguishing between low and high to maximize this positional information. So this is the most you can get. So saying like you know the input signals, and now you just, the task is to define the thresholds in the way that they maximize this mutual information or positional information. And you don't get even, you are, let's say, close to 3 bits. So it means that the cells in the actual Drosophila, they are using also intermediate states. So it's just, with just binary states, it wouldn't work. And now I will stop with the Drosophila, which I think is, it's well studied, and you can do many interesting things, and we'll now go to the stuff I'm doing, and that is studying spinal cord formation. So this is something I started as a postdoc, and then I continued now as in Krakow. And the main, there are many differences between those two systems, but just to give you an overview of what happens here, you have the spinal cord and you take the dorsoventral cross-section. On the ventral side, you have the source of Sonic Hedgehog, a morphogen. This morphogen spreads across the tissue, forming gradients of concentration. On the dorsal side, this is BMP, another morphogen that is spread from the roof plate, also forming gradients of concentration. And these morphogens are interpreted by, the morphogens are interpreted by cells, by gene regulatory network, resulting in the stripe pattern of gene expression domains that are different neural progenitors. And these neural progenitors will later on give rise to different types of neurons. So the fact that I'm able to, move my hands, the fact that I'm feeling, heat, and it's because these different stripes are formed in the proper way. But there is more to this picture, and especially if you compare with Drosophila, where things happen quite quickly, the embryo there is not extending in size a lot. Here, you actually have a lot of growth. So the tissue, the spinal cord, it extends from 100 micrometers to 400 micrometers over 60 hours in this dorsoventral extension. And what you see here are the expression of some target genes and some signal and morphogene signaling activity. So to get this, my collaborator Anna Kicheva, she took 300 spinal cord cross sections taken at different embryonic days. Those were immunostained to mark the expression of these target genes and signaling activity. And now if we quantify this signaling activity, what we see is that for BMP signal, initially they are quite close or very close to exponentially decaying profiles. Over time, and here we represent it in relative units, so we take the position in the tissue and divide by the length of the tissue, they effectively move away from each other, which is just saying, we had in the previous that when you have diffusion, you get the characteristic decay length. So this is almost the same throughout this development, but because simply the tissue is getting larger, the relative range of these morphogens is shorter. But the story is different for the target genes. So here are some examples, PAX3, NKX6.1, dorsal ventral side. Initially, they are highly correlated with the input signals, but over time, especially after the morphogens move away from the center, you see the formation of the sharp boundaries. So they get this sigmoidal shape. And another way to represent that would be if now instead of having this relative position, we have absolute units. And so you can imagine that, or imagine like here there is time, here there is space, the tissue grows, your spinal cord extends, and then the BMP signal stays on the dorsal, and on the ventral, there is some change in the amplitude of the signal, but in the decay length not so much. You get this point around 30 hours where there is nothing there, or almost nothing. And this is different qualitatively in the target genes. So how can we use that? Let's see. First, we wanted to quantify. Yes. Maybe I didn't hear the first part.
In this case also, are there sources at the two ends for the BMP signal and the sonic hedgehog?
Yes, yes. The sources are at both sides. For sonic hedgehog, this is floor plate, and on the dorsal side, this is roof plate. But it's actually very also good question. How the source size and how it changes with this. And I will address that on Thursday. But just to give you a brief idea, the source is also extending with the tissue, but for the floor plate, it's actually extending slightly slower than the rest of the tissue. But yeah, that's something we're also investigating. But yeah, the sources are there. So they are constantly, producing, but yeah. So first, we wanted to quantify this positional error. So again, just a reminder, if you would have a single signal, you just get the variation in your gene expression, and then you get the positional error, the uncertainty. But because we had, or you can do it for one signal, like PSMAD is the effector, so you have a signaling pathway. So that's actually our readout for BMP. This is our readout GLI binding site tagged with GFP for sonic hedgehog signal. So you see that just one morphogen would be enough to specify with 3-cell precision of so this positional error of 3 cells, only just the dorsal side. The other one would be mostly fine in the ventral side, maybe somehow worse in the middle. But if you have combination of both, then you need to extend this definition of positional error. But you roughly end up having something that a system where two morphogens could early on, this is time, so if for light shaded, we had the cells in the tissue, the target genes are specified, or okay, not yet the target genes, but the input signal, so the morphogens are capable of specifying the cell fate with this ±3 cell diameters, and this is done uniformly across the tissue. And now if you look into target genes, okay, this is the position of their boundary. There is some maybe, there are different reasons why it shifts a bit, but if we quantify the variance of this in precision, we see it's also on the order of this ±3 cell diameters. So we asked whether this is just coincidence or maybe the cells are doing also this optimal decoding. And we tried to explore this framework. This paper also, Gasper is on that one, using just two morphogen signals, saying like BMP and sonic hedgehog, and okay, the rest is exactly the same, so I'm not going to explain that. We apply Bayes' rule, and we don't even want to construct the decoding map as defined previously, but we want to get this posterior distribution. So given my input signal, what's the most likely fate? And represent it also as a kind of, decoding map, but meaning this posterior distribution. So that's the nomenclature we used in this paper. So here what you see is that now the cell fates are decoded with different colors. If the cell is predicted to have dorsal fate, it's red. If it's predicted to have some intermediate fate, then it's white, and for ventral, you get something blue. And this black line is the prediction for the wild type, or no, it's what is measured in the wild type. And why you see a straight line? We have log-log plot, and we had exponential gradients on both sides. So if you have both, exponential concentrations, and then you do the log-log, you end up with a straight line, and then this prediction for the wild type works really well. But this is how we formally define it, and we like this implied fate to be the just X that maximizes this probability that is defined here. And now with this, we could move a bit farther from this wild type line and see whether we could make predictions for mutants. So one prediction was if we now have a hypomorph mutants where the soy hedgehog is reduced, you get shift in the gene expression domains, saying like the ventral domains are compressed and the intermediate domains are broadened, and that was the prediction that we could also compare with the experiment. But the interesting part was this shaded region here that I didn't explain what it is. And why was it interesting? Because if you now look into not the most likely fate, but you look into the distribution of these fates, you see that in this shaded region is actually getting bimodal. So here along, let's say, wild type, it's kind of sharply peaked. Here is a bit more broad, but here is bimodal. And now if I replot it in terms of specific cell fates, the prediction would be that if we take the tissue and expose it to high concentration of both morphogens, we could see a mixture of dorsal and ventral fates. And I was very excited about that. You do your theory, you work a lot, and then this is like genuine prediction that tells that if you now take the tissue explant and you expose it to this high concentration, you would get a mixture of cell fates. And that's what we observed. So how you see it? So this is chick explant tissue from the neural tube that was exposed to high concentration of BMP, high concentration of snake hedgehog, and you don't see, and there are different markers here. So red cells mean dorsal fates, green cells mean intermediate, and violet cells mean ventral fates. But you don't, so the thing is you don't see green cells either here or here. I don't have one with the two of them together, but they cannot be also expressed in the same cell. So that was kind of very convincing and gratifying that the cells are actually, uncertain about their fate. So they either become dorsal or ventral, and one could also reproduce the rest of this decoding map. And again, I'm not going to explain that in details, but if you have high concentration here in nanomolars, you see the ventral fates, and basically no other fates. And then this was something that I already showed, and this is something showing intermediate. So, okay, here you don't see really well, but if you quantify it, there are green cells here and here. And now, I used this almost an hour to explain, how we can do this optimal decoding and so on. But is it really like can gene regulatory network do that? So that was the question, because we know how that we can apply, we can kind of have the input, we can have the output, we can have a theory for optimal processing, and then we can make predictions. But can we construct a gene regulatory network that is actually doing this task? So I propose a 3-node network composed of key transcription factors in the neural tube, like representing ventral, intermediate, and dorsal fates. And then this, each of them were like inhibiting each other, like to some extent, or the core network here would resemble the toggle trad that we heard yesterday. But here you additionally have input signals from this morphogen activity like Swing Hedgehog, BMP, and then there are also uniform activations. And then you define the dynamics of the system through thermodynamical model, where this is kind of standard. You have some terms. If you have inhibition, it's it's higher level of the strength of inhibitor makes the whole thing smaller. So and these are the Hill coefficients. And yeah, if you want this to be more like classified, like we had some parameters that we were perturbing concerning the strength of interactions in the network, and we also had some parameters that were fixed during the screen because we wanted to see whether we could find such a networks. So how to do it efficiently? So here we didn't do anything fancy. We rather, or I did some brute force approach. So I was testing, or wrote the code that was testing 10 to the 8 sets of parameters, or these 13 parameters, and then we were constraining that to see how many of those produce three-stripe pattern. So out of this 10 to the 8, 10 to the 6 produce the right spatial pattern, then the temporal pattern, because we knew that there are these shifts of the gene expression. And finally, roughly 1,000 of these parameter sets produce the decoding map that has these characteristics that for the high concentration you have either ventral or dorsal fade. And okay, you can do different things with this set of successful solutions, but one thing we did, we applied principal component analysis to see whether they are somehow different or they might mark different regions in the space, but that wasn't the case. They rather create a single cluster where you could quite well interpolate between different successful solutions. And finally, we wanted to check whether the successful solutions are able to reproduce the gene expression boundaries with the same precision or positional error that we had in the actual data. So we took the variability from the actual data and provided this as an input. So we had this sonic hedgehog and BMP signals fluctuating, and what we saw is that different networks provide a different level of this imprecision. I didn't mark here the biological, but you remember this plus minus 3 cell diameters. So it's somewhere here. So we could see that most of the networks could get there, but a few of the networks could get there, but most of them are actually doing worse. But it's still like we are in this range of biological parameters, and if you would say that there is some evolutionary pressure for that, and maybe we missed some of the constraints because we didn't put a constraint on, apart from the decoding map, there was nothing telling these networks to just minimize this positional error. So we were kind of happy to have at least a few that are in this biological range. And yeah, I'm slowly getting to the conclusion. Just I want to sum up this two parts of the talk with slides or with a figure that I borrowed. And this is very recent and quite very well written summary of the review paper by Kachin and Gregor in Development that summarizes their contribution, their and other authors' contribution to this framework of positional information. So I highly recommend that one. And the idea is about how we understand the flow of this positional information during development. So there is some signal that encodes the positional information, but first you need to establish this gradient, and this is we had this in the last talk. Like there's some maternal messenger RNA that codes for bicoid, and then this is through diffusion and degradation makes a gradient, exponentially decaying gradient in the space that is then read out by the cells. So you could use this definition of mutual information that I provided you before, like if you get the total entropy of the distribution of the signal, subtract the noise that is there, you end up with a useful part. This is mutual information. And for every such signal, input signal, you could get here for bicoid, you could get this mutual information. And now, and that's another, maybe an open question. Partly I would like to discuss this also during tutorial on Friday. Like, this is then recoded. Why is it recoded? We don't know. Like, you could say maybe it provides you easier interpretation of the signal, maybe better representation, maybe cells could somehow, this is relatively simple compared to this shape, okay? So maybe this shape in combination with other shapes gives cells like more robust, maybe just more easier to implement way of decoding the signal. So there's this type of recoding that can involve multiple steps. It can happen many times during development. And then here in this first part of the talk, I focus on presenting basically this approach on how you get the hunchback and other gap genes, the positional information in hunchback about the cell fate or the implied position, decoded that to get the optimal decoding and then to specify the pattern. But like in the second part of the talk, I showed that you could use that in the different system and construct a gene regulator network that is actually doing this decoding. So this framework, if you think about that, it's broader. You don't need to apply it to biological signals. It can be any kind of signals, and it's just enough to have the proper definition of the input-output signal, and then you apply the well-known bias rule. And again, just to summarize and maybe highlight some of the open questions that are there. I would say for me, it's one of the things is that you don't, one could expect that many of the organisms that were, basically we believe are well shaped by evolution, but how much this theoretically optimal patterning is under selection pressure, saying like, I can imagine cases where this is indeed needed, like in maybe Drosophila, you want to use the available nutrients and everything to really maximally use this information. This might help you to be. This gives you the fitness advantage, the selection advantage. But there might be cases where you don't need to, maybe, you don't need to invest so much into designing, constructing something that interprets the positional information optimally, but rather the thing is focused on the growth. Just you want to grow the organ. I don't know, in the heart, the pacemaker region, it was somewhere mentioned. The interesting fact is the pacemaker region, the cells there proliferate slower than in the rest of the heart, like chamber. They proliferate faster. And also, I know another example, cancer. Okay, this is, in cancer we heard that the thing is growing, but it doesn't, the cancer, as far as I know, it doesn't care about precision, nothing. Just want to multiply, but again, that wasn't, shaped or optimized by evolution, or at least, for for our and other animals, yeah, selective fitness. The other thing is, and it also appeared in the last talk, someone asked about, is the temporal dynamics matters? Does it matter? So here in this picture, we had basically static picture, which means it's not necessarily, you know, completely static, because these steps, decoding, they need to take some time. So there is some temporal window when the signal is decoded, and then when it's, you know, it's established at the end. So what was, for me, very interesting in the spinal cord is that you actually need just the first 10 hours to specify something later on at 60 hours. You could, if you do this decoding later on, you don't have so much information. And similar things are in Drosophila, saying like this kind of works quite efficiently, saying like very early time points are often enough to specify pattern that is established later on. Of course, there is a related question: is positional information produced during development? So I mentioned this data processing, you know, inequality, like you lose information when you process it, but I can also give examples, like Bicoid has something like, I don't know, 1.5 bits, and Hunchback on itself has 2-point-something bits. So how is it possible? And the answer is the cells are performing this spatial temporal averaging, so they use more of the signal than you just would have from a single snapshot. So there is a bit more, and this is still open question, how to do it properly in the mathematical framework. Yeah, another related question, this recoding, how different structures are related to representing positional information. We work a lot on different gene regulatory networks, but the question here would be what kind of gene regulatory architecture may be most effective in decoding. This we don't know. Or what would be, if you think about robustness of positional information, you can think about different sources of variability. I mean, like there is, okay, noise, like intrinsic noise, but then also there are some embryo to embryo variability. How much of that is actually variability that is kind of converted with this framework of positional information to the output signal, or how much of it is actually not relevant? This is another open question. And one more that I listed here is how this positional information framework is related to cell fate or canalization, because up to now in the second part of the talk, I made these different colors. So I did, I discretize my continuous readout, because previously those were the position, implied position, which was just continuous. But of course, cells in the tissue, they have specific cell fates. Another question is, what is cell fate? And you know, how this is related to position, saying like different expression, but that's also question, that maybe should be answered by people doing biology. You know, what defines the specific cell fate? But the thing is how to adapt this scheme to not having the continuous readout, but rather some discrete readout of position. Yeah, and finally, an open question: is it how general this optimal decoding of morphogenesis, is it a fundamental principle that can be identified in different developing organisms? Yeah, the time will tell. And yeah, I would like to thank. Okay, it's here. I didn't present a lot of research, of my research. Just the second part was done with people from IST, Anna Kicheva, from James Briscoe, Tobias Bollenbach, Gasper Tkaczyk. But yeah, like I use that to have to others plenty more questions. And this I will discuss on Thursday also, how this whole picture is affected by tissue mechanics, how this picture is affected by the source formation. So this all led me to meeting great people, and they are yeah, listed here, current and past members. And yeah, we also always look for students interested in joining the group and postdocs. So yeah, if you want to move to Poland, the temperatures are moderate. So yeah, you are welcome. Thank you.
One of the problems, there were these two morphogen gradients from both ends, and you were talking about the scenario where both the strengths are, both the morphogens are upregulated. I mean, the strength of the morphogen is very high, and that would lead to mixture of these sulfates, right?
You mean like here, the arrows are, you mean they are upregulating the genes? This is what you mean? No.
But before the gene regulatory part.
Okay.
Yeah, this one. So the black line corresponds to wild type, right?
The black curve corresponds to wild type, yes, and maybe here it's more visible, saying like, it's simply saying that on one end you have, let's say in the ventral end, you have high concentration of the ventral morphogens, also in hedgehog, and low concentration of BMP. And if you put two exponentials and do it in the log plot, then you get this line that you know ends up at something that is high BMP and then low sonic hedgehog activation. And then here this is for idealized profile, saying like we fit, we get the amplitude, and we also get the decay length. So this is idealized, but I don't have it in this presentation, but one could also produce I think it should be in the paper, in the supplementary materials, the map without any idealized profile, saying like from the raw data, saying like you just construct your, from the data you get the average signaling activity, and then you construct the decoding map. You basically need the average of the signal and the variance, or in general, you know, the noise, but if you have more, then you need the covariance matrix. And here we saw that basically the covariance terms is negligible, saying like at least in this case.
So for the gene regulatory motifs that you've sorted out through various filters,
you mean this? Sorry.
Yeah. Finally, you showed that there are some cases where the precision can go to realistic values, right? So
yes.
Could you have an analogous map for those circuits, compare it with the wild type?
So it's a very good question, and I did some follow up on this, saying like, or the way I understand the question, can I, you know, somehow distinguish between different topologies or network motifs, you know, resulting in different imprecisions? So I would say in the end I didn't have a clear answer, saying, you know, what's the cause, but one part of the answer was you could distinguish some of the interactions were stronger than the rest, but it wasn't like a single. You know, single interaction was decisive. It was rather a combination of some of the interactions, but it wasn't very clear. Saying like, I also talked with Jolly about, you know, toggle trad and so on, and like yesterday, not back then, but it's not like there is. It's rather a combination of parameters that would decide, but I didn't follow this. So, you know, it's still like an open question what makes, even in this particular system, what properties of the network would result in different patterning precision. Yes?
And these would be sort of effective rather than the direct gene interactions, right? This would be an effective interaction.
To some extent, yes, because the whole system is more complicated. But also, it's not like you can think about some subnetwork of the larger network with these nodes. They have their names, and 5 out of these 6 interactions were already identified in the literature. We also showed that you could get the 6th one simply by simulating all other combinations with just 5. And couldn't reproduce all the constraints that we had. So that was kind of a way to get this 6th interaction. I don't remember which one was that, but yeah, it's also in this. Yeah, you can look into works of James Briscoe and his lab where they studied other networks in this ventral, also a bit larger networks, and there are lots of toggle switches there. As we heard like yesterday, when you think about development, and here this spinal cord development is really about fixing fates of neural progenitors or cells that will later give rise to different types of neurons. You want to specify them and let them stay that before they move to the next stage. So they maturate to become neurons. So yeah, there are lots of toggle switches there.
Yeah, so you mentioned about this embryo to embryo variability can also like change the position information that can be gained by a cell, right? So I mean, what I think of like embryo, when you change the embryo, you are changing the environment sort of. So it is changing the, like, it is like the extrinsic noise is coming into play. Whereas mutual information is only dependent on the intrinsic noise, right, in the system.
Yeah, so it was more like an open question asking, yeah, to somehow rephrasing what you say, you know, like, what's the, let's say you do experiments, you do it in some specific conditions, but then in nature, in more, you know, in real environment, you somehow have different fluctuations, I don't know, of nutrients, of temperature, and so on. So how you should include that into this picture? And this is like, you know, a bit, yeah, because it could cause different, you know, shifts in this, in the input signals.
But that noise entropy is due to intrinsic noise.
Yes, or yeah, the way in this study, yes,
Yeah.
you know, it's very well controlled, and especially like Thomas Gregor, like I think in 2007 in Cell, they had a paper on different sources of noise, and it's quite, I would say, still very relevant thing, like, because you want to measure this noise, and you want to minimize any other noise that is like caused by, you know, the way you do the measurement, the way you, I don't know, stain, the way you do the cutting, and so on, so on. So they identified these different sources, and they were very confident in that what they are measuring is indeed this intrinsic noise, because you also want to have everything well controlled. So yes, saying like this, so short answer, this is intrinsic noise.
And just one more question, like that in one of the slide, there was this inverse probability distribution. It had this bimodal nature. So why, in which case it might happen, like that?
So that's, you know, the thing is, in biology, in the wild type, it probably never happens. Saying like, that's why, you know, as a person doing theory, I was kind of, because the relevant concentrations are here. relevant for the wild type. The wild type experiences that. Maybe in one case, you know, you would, in the mutant case, you would lose one of the gradients, but it would basically mean, you know, you shift this curve towards this, or this towards this, maybe some upregulation, but so it was in a way really, you know, bold prediction to ask whether this has something to do with biology, but apparently it has. Saying like it might also inquire some maybe deeper question on if you just have the wild type here and then you would construct networks here, would they produce this? And not all networks do it. This we saw in the screen, but some of them do it, and also that's something we observed experimentally. So yeah, you can imagine also different ways of decoding, and I'm not saying, you know, this is like super important. Saying like for people doing biology, and this probably for them this is important, and the network that can make their predictions. But I'm also, you know, sometimes you need to, yeah, explore what's possible, and that's one of the results that, you know, you just find this interesting possibility, and then you could test it experimentally, and it turned out to be correct.
Oh, thank you.
So Marcin, I actually had a, well, I don't know what a question, but you know, some of the things you mentioned actually has a very curious parallels with the problem of how, for example, the cell is able to detect a signal from noise, its receptors, because, you know, it's this constant amount of, you know, stimuli which are bombarding a cell. So how does it know a relevant signal from noise? And like, for example, when its signaling network is, let's say, has something binding to the upstream receptor, so it has to figure out whether this is noise or not. And so this Bayesian approach is also used over there. So I'm just curious, like, you know, there's a strange parallels that I see. So do you think that there is some kind of a universal principle that is operating both at the level of the cell, trying to make sense whether what it's binding to its receptor is signal or just something it should ignore, and also like from the question of, you know, what exactly is the concentration of the morphogen, you know, given that there's a lot of fluctuations going on?
Mm-hmm. Yeah, so you know, excellent comment. And first, to the first part of that question, I would say this whole concept of information encoding then decoding is somehow agnostic to you know what the, you know, it's in the end you could think about this network and all related like receptor signaling layers as a black box, you know, and you just take what is at the start, you just take what is at the end, and you know, you say, having that, can we make a prediction? And in the Drosophila or spinal cord, the answer is yes, you can. So, but that brings, you know, the next question, you know, how the cell is doing that, and that's actually the, you know, for me also a key question, how the cell is processing this this signal. So my personal opinion on that is that when you have these different layers of, you know, receptors signaling, they don't care about, you know, what is noise, what is not. It's just interpreting it, and at the end of the day, you know, you might have these different layers that get read out of, let's say, it's hard to say whether they get read. They just process it to have a different representation. That I would say the noise is there, but at the end, whether it's making like a switch-like behavior or it's based on compiling larger number of signals into a cell fate, yeah, I would say it's for me it's also like you work with the ODEs, so you have some system, you put some input, and you get a solution, you know, which okay, without noise, this is just deterministic. When you add noise, you have this stochastic component, but and you always end up also with variability in biology, saying like, so I would say this noise is there, saying like the cells are, you know, they can decide about what decide. They can specify their fate with some precision, but it also means they can make error with some probability. So it's yeah, I would say it's, yeah, you could think about this, you know, from both sides, saying like either I specify precise pattern, but still, or very imprecise. So it's always given by this probability. So this noise thing is always there, even at the output. Yeah, and the other one, the second part of the question, can you repeat? Like.
Yeah, so do you see any parallels between the way the the cell is able to, in some sense, figure out that no, it should ignore this particular?
Oh yeah, but this I think you know, somehow if that's I don't think there is like a parallel. I would say it's still processing all that, and at the end you just get sometimes you get errors. But you know, from design perspective, maybe you would try to minimize that, and to get architecture of the network the way it you know, minimizes this position error. That's I would say what, yeah, what, and there are also trials that, yeah, I think right now, I'm not sure whether it's on a bioarchive, but a follow-up of this. So Thomas Sokołowski and Gasper Tkaczyk and other, they also try to, using this constraint, you want to construct the network from like ab initio, saying like from just having this constraint, you want to construct the network, and whether you end up with something similar. So I know that they were working on something like this for the full network, saying like providing this black box, yeah, example, and I think that's either already submitted somewhere, or I think it's submitted somewhere. And for what I thought with Thomas, it worked, saying like you can make prediction about, of course you get other networks, other architectures, but the one that you get is also consistent with what you observe experimentally. So yeah, it means that maybe this principle is kind of really general, or can give you this ability to construct something from the scratch.
Okay, if there are no further questions, let's thank Marcin once more. Okay, so the last activity is actually the informal group project discussion. So we start